Hello, welcome back to Introduction to Genetics and Evolution. In the previous videos, we talked about some of the fundamentals associated with natural selection. I reintroduced that it is a mathematical inevitability. We talked about the three conditions for it, and we looked briefly at changes in allele frequencies and genotype frequencies associated with the action of natural selection. Now, one point I stressed in the last video was the importance of dominance. The dominance affects how natural selection shapes allele and genotype frequency. Now, in the last example I showed you in the last video, we had all adult little a individuals die. So we said that at age 10, all little individuals just die off. Let's follow up on this with a couple of different examples. I'm going to change, in this case, the alleles from big A and little a to M and N. And that way there's no implication of capital being dominant or recessive or anything like that. So let's say now we're looking at the relative fitness of three possible genotypes. And again, there's two alleles. Genotype MM has a fitness of 1.0. This is again relative fitness. Genotype MN has a relative fitness of 1.0. And genotype NN has a relative fitness of 0. As in all individuals that are NN are dead. Now, which allele is dominant? Which allele is recessive? Well, we define dominance by what the heterozygote has the same phenotype as. The phenotype we're looking at in this case is the relative fitness. So which homozygote does the heterozygote have the same relative fitness as? In this case, the MM homozygotes have the same relative fitness as the MN heterozygotes. In this case, this must necessarily be dominant, this must necessarily be recessive. Okay? Because these two have the same fitness, this one must be dominant, the M allele. This one is recessive. In this particular case, the recessive one is also bad. This is the one that has the lower fitness too. That won't always be true, but in this particular example, that is true. So N is recessive to M, and N is detrimental or bad. Here's a different example. So in this case, MM has relative fitness 1.0, MN has relative fitness of 0, and NN has relative fitness of 0. So what do we think is going to happen in this case? Well, first of all, which one is dominant? In this case, again, we look at the heterozygote. Which one has the same relative fitness as it? In this case, it is the NN homozygote. So N is actually dominant now. N is still bad. So in this case, the dominant one, this one is dominant, N is dominant. And what will selection do differently? Well, we might guess in this case that selection will actually be much more quick. Because in this case, you're eliminating all N's from the population at once, instead of just a subset of N's from the population. Here, some N's are sheltered. Here, no N's are sheltered. They are all exposed to natural selection. So, with a dominant detrimental, the heterozygotes will also respond to selection. Okay? Now let's do a third example. Here we have MM is 1.0 for its relative fitness, MN is 0.5, and NN is 0. In which case, which one is dominant? Well, the simple answer is neither, because no homozygote has the same relative fitness as this particular heterozygote. So we can call this a case of no dominance. In all three of these cases, we assume that the NN heterozygote is the worst one. This is what's often referred to as directional selection, because selection will preferentially push towards the elimination of the N allele over time, whereas the MM is always perfectly healthy. But the rate will be very different. In this case, selection would be very slow to eliminate N's. In this case, selection would be very fast to eliminate N's. In this case, it would be intermediate. Now, we can see this. Here is the effect of dominance with directional selection. This is using that allele A1 software that I mentioned before. So let's look at the case now. The alleles in this case are called A1 and A2. So if we have allele genotype A1A1 has a fitness of 1.0, genotype A1A2 has a fitness of 0.5, so we're not eliminating all of them, we're just eliminating half of them. It has half the relative fitness. Genotype A2A2 has a fitness of 0.5. This would be a case of a dominant detrimental. We say it's dominant because, again, you look at the heterozygote, has the same fitness as this one. Right? So A2 is dominant, A2 is bad. A2 will be going away from the population. In this case, since no A2s can hide, it goes away pretty quickly. In contrast, when you have the recessive detrimental, now this one is dominant, A1 is dominant, but A2 is the bad one. So you can eliminate these pretty readily from the population, but these are perfectly okay. So as you can see what happens here, if you look at the change over time, the x-axis here is time in generations. The y-axis is the frequency of the A2 allele. You see the A2 allele is going down in frequency, but it's going down somewhat gradually as it gets near the end, because a lot of these individuals, when you're down here, a lot of the A2s are hiding as A1, A2. Whereas over here, none of them can hide. They're immediately exposed to selection. Boom. Whereas here, the A2s can stick around, and therefore you maintain some A2 in the population over a fairly long period of time. Now, in fact, this is actually what happens for many bad mutations. As I mentioned a long time ago when we were talking about inbreeding depression, many bad mutations are either completely or mostly recessive. Some examples of genetic diseases like this are Tay-Sachs, cystic fibrosis, etc. They're maintained in the population by carriers. Selection is inefficient at getting rid of them. Now let me give you a problem to try. Which of these depicts the change in lactose intolerance allele? This is the big A that we talked about in the previous lecture by selection. So I've given you here the relative fitnesses. So big A has a relative fitness of 0.95. That is when it's lactose intolerant. That's also the ancestral type. Big A little a has a relative fitness of 1.0. And little a has a frequency of 1.0. Or I'm sorry, a fitness of 1.0. Again, these pictures are showing time and generations on the x-axis and the frequency of big A, make sure you know that, the frequency of big A on the y-axis. Which of these would be an accurate depiction of what we expect to see over time? Well, I hope that wasn't too difficult. Now, what do we have here? Now, which allele is dominant? That's the first question to ask yourself. Well, the allele that's dominant in this case would be little a, because it has the heterozygote has the same relative fitness as the little a homozygote. So this one is recessive. Big A is recessive, and it's also bad. So we have a case of a recessive detrimental that's being eliminated from population. What we're looking at in this case is the frequency of big A. So that is the recessive bad allele. Since it's bad, we know it'll go down in frequency. So we can immediately eliminate these two. Since we know that big A is bad, it should go down in frequency, not up in frequency. Now the question is, does it go down fairly quickly, or does it do this very slow thing? Well, as I mentioned in the last slide, when it's recessive, a lot of individuals here can hide as big A, little a, and it'll be slow. So in this particular case, number 1 is the correct answer. Now again, there are many types of selection that can happen. And there's in fact many types of what is referred to as directional selection. And this is what we've been looking at so far. In directional selection, one allele eventually replaces the other. That everybody in the population eventually, if you wait long enough time, will be either big A, or little a, little a. So what you have is something like this, where the fitness of big A is less than or equal to the fitness of big A, little a, is less than or equal to the fitness of little a, little a. Or the opposite, where the fitness of big A is greater than or equal to the fitness of big A, little a, is greater than or equal to the fitness of little a, little a. Now the examples can vary. So here we have a case where we have a recessive detrimental for the first line. You see this because the dominant one has the high fitness, right? The recessive one. So eventually all the population will be big A. All right, here, what's happening eventually. Okay. In this particular example, again, everybody eventually will be big A. This is a case of no dominance, or intermediate dominance, as some people call it. In this particular example now, we have little a has higher fitness than the other two. So eventually everybody in the population will be little a. And here's the example of lactase persistence or lactose intolerance, that eventually everybody in the world, if we wait long enough periods of time, everybody should be little a, or lactose tolerant, or lactase persistent. Now this may not happen in real life for this particular example because we now can purchase lactase over the counter, but that's the direction that things have been. So these are all cases of directional selection where eventually you go to one form. You eventually go to monomorphism. Here is a different type of selection. This is the case of heterozygote advantage. This is often called overdominance. In that particular case, the most fit genotype is the heterozygote, or the big A, little a. So this one is more fit than both kinds of homozygotes. The two homozygotes may differ in fitness from each other, but the highest fitness is associated with the heterozygote. Okay? In this case, unlike with directional selection, one allele does not replace the other. This does not lead to monomorphism over time. The classic example of this that people always cite is that of sickle cell anemia and malaria resistance. So let me introduce this to you briefly. Malaria is a big threat for a large part of the developing world, In fact, in places like sub-Saharan Africa, your odds of death by malaria are around 4%. That's pretty high overall. Now, malaria is transmitted by mosquito bites, which are giving this plasmodium protozoan, which is what actually causes malaria. Now, how does this relate to sickle cell anemia? Well, sickle cell anemia is not a great thing either. This is a recessive genetic disease, so only little a individuals are fully afflicted. In this particular case, the sickle cells die faster than normal red blood cells, and they deliver less oxygen to cells. So people who have this disease are tired all the time. They have chronic pain. They have quote unquote episodes where they sometimes have to go to the hospital associated with this. So this is clearly not a good thing. Interestingly, the heterozygote, so the big A little a's, these people are often called having the sickle cell trait. They're usually okay. Sometimes they can have some sickling of their cells when they've exercised very hard. They've had some sort of intense physical exertion. But overall, they're good. But interestingly, these individuals that are heterozygous for sickle cell are actually more resistant to malaria than individuals that are homozygous for the non-sickling allele. Now, the thought a long time ago of this was that invasion, growth, and development of plasmodium was actually somehow reduced in these heterozygotes blood cells. But more recently, I cite two studies down here. You're welcome to look them up if you'd like. One thought is the heterozygote is more tolerant to sickle cell symptoms, but actually still retains infection. Whereas another study showed that the infected heterozygote cells are more likely to be eliminated by the spleen. So there are a couple of different explanations for this, but the important thing is that if you're in a place where there is malaria, if you're a heterozygote, you are actually not immune, but you are much less likely to get it than individuals that don't have sickle cell trait. So the sickle cell exhibits a heterozygote advantage in some populations, because you can think of it this way. The big A's are susceptible to malaria, so they might have a relative fitness like 0.85. I'm just making up these numbers in this case. The big A little a's are generally fine, right? They don't really get malaria. They don't have sickle cell disease, so they'll have the highest relative fitness, 1.0. And little a's have the sickle cell anemia disease, which is extremely debilitating. So we'll say their relative fitness is something like 0.05. Well, what would be the fate of the little a allele if it arose as a mutation in a big A population? Well, it seems like little a is really bad. On the other hand, big A little a is pretty good. So what should happen? Well, we can simulate this using allele A1, and we see that the frequency of little a rises and then comes to this equilibrium. It comes to this stable allele frequency, and it just stays there. Now, the number it comes up with in this case is the frequency of little a, or A2 in this case, is 0.136. That's what was estimated using allele A1. Now, interestingly, we can actually apply the math ourselves and see what happens. So the frequency of little a, this is the prediction from what you should see in terms of changes in allele frequencies in the equilibrium allele frequency. So this is at equilibrium. Put a little hat over this to indicate equilibrium. At equilibrium, what should the frequency of little a be? Well, we'd say it's 1 minus the relative fitness of big A, divided by 1 minus the relative fitness of little a, plus 1 minus the relative fitness of big A. Well, we have all these numbers here, right? So the fitness of big A, this one is 0.85. The fitness of little a, 0.05. The fitness of big A again is 0.85. So we can substitute these numbers into this very simple formula. So there we go. They're put in there. And this calculates out to 0.136. That number may look familiar to you because that was exactly where we saw this equilibrium drawn here. So the important thing, this is actually a stable equilibrium. That essentially, if you're not at the equilibrium allele frequency, selection pushes you towards it. If you're above it, you go down to it. If you're below it, you go up to it. You're drawn to this equilibrium allele frequency. And again, this is called overdominance. Both alleles are retained, and if you're not at the equilibrium frequency, you will move to it. Now let me show you a graphical representation of that. Here it is. Here's a graphical representation of changes in allele frequency associated with overdominance. On the x-axis here, I have the frequency of the big A allele, and on the y-axis, I have the predicted change in frequency of the big A allele next generation. Don't worry about this too much if you're not exactly following, but this just depicts the same sort of thing. So imagine that you're at, let's say in this case, the predicted allele frequency, equilibrium allele frequency is 0.6. If you're at 0.4, if you're at the actual frequency of 0.4, your predicted change in frequency of big A is what? In this case, your predicted change is positive. So I'll push you towards the equilibrium. If you're over here at 0.8, your predicted change in allele frequency is negative. So that pushes you in this direction. So you can see anytime you're anywhere but at the equilibrium frequency, or where you have no variation, then you will always be drawn to that equilibrium, unless you're at either 1.0 or 0.0 where there's no variation. Again, selection requires variation to act. So this is just a graphical representation of what I told you in words, so don't worry about that. Again, the important thing with heterozygote advantage or overdominance is there is this predictable equilibrium allele frequency that the population will be drawn to. Now let's talk about the opposite, heterozygote disadvantage. What happens if the heterozygote has the lowest fitness? There aren't very many good cases of this, but we can at least show mathematically what should be expected. Well, in this case, again, the least fit genotype is the heterozygote, so I just made up some numbers here. And what this does, this actually leads to an unstable equilibrium, kind of like this fellow standing on the chair here. That if you start below the equilibrium allele frequency, so again, the equilibrium allele frequency is 0.27, so you're just a tiny bit below it there. If you start below it, you just go away completely, and you lose one allele completely. If you start above it, you lose the other allele, and one allele goes to fixation. It's only if you're right at the equilibrium allele frequency that both alleles can persist. But in a real population, you probably can't do that, because unless you have an infinite population size, you can't stay exactly at that allele frequency. So unlike the previous example, this is a graphical representation of allele frequency change with underdominance. If you're not at the equilibrium point, let's say, for example, again, the equilibrium allele frequency is 0.6. If you're starting at 0.8, again, your predicted change allele frequency is positive. You get pushed away. If you start at 0.4, your allele frequency predicted change is negative. If you're negative, you get pushed away. So again, you lose variation in the population. Only if you're right at that equilibrium allele frequency can it stay. So it's a very unstable equilibrium point. It probably doesn't happen very much, or if it does, it's eliminated so quickly that we never see it. Okay? So there's the three kinds of single locus selection we've talked about. Directional selection, where one allele is just consistently favored. Overdominance, where variation is retained in a stable way. Underdominance, where variation can be retained temporarily in an unstable way, but eventually you lose variation, and it's unpredictable which one you would lose unless you know that you're above or below the equilibrium allele frequency. There's one more type I was going to introduce, and that is that of frequency-dependent selection, and specifically negative frequency-dependent selection. Now, the previous examples all assume that fitness was independent of what was going on in the rest of the population. That, you're big A, you're better than big A, little a, no matter what, right? Or if you're a little a, you're better than big A, no matter what. Sometimes it's actually better to be rare, that your fitness may depend on your relative abundance or frequency in the population. Now, being better makes you more common. Right? That if you're better, it means you're having more kids, you're more likely to survive, so you become more common. But if it's better to be rare, it's kind of this funny thing where you have these opposing forces. What this does, and this is the case specifically of negative frequency-dependent selection. We're not going to talk about positive frequency-dependent selection. If you have negative frequency-dependent selection, this assumes that it's better to be rare, but being better makes you become more common. This eventually leads to an equilibrium. Let me give you an example. Let's use the case of sex ratio. So in many species, sex is determined genetically, as you know. That in mammals, we talked about the sex chromosomes, that XY is what causes maleness. You're kind of locked into this by transmission. But there are other species where alleles at one gene can cause an individual to become a male versus a female. That you can have this sort of variation within it. Now, if females are very rare in the population, and you need both males and females to make offspring, is it better for you to produce male or female offspring? Is it better to produce the abundant type? Or is it better to produce the rare type? Essentially, in this particular example, would selection favor the male or the female allele? It's probably better to produce the rare type because you're more likely to mate. If you produce the common type, you're very likely not to mate. So let's use kind of a silly example. I did a little bit of Google searching on the web, and I tried to find a picture which showed a very skewed sex ratio. So one of the things I typed in was computer science club. And this is the picture I came up with, which happens to have a lot of guys and one female. Now let's imagine this computer science club was marooned on an island. Okay, what is the likely fitness of the average of all these guys in the population? Well, it's probably pretty low because probably they're not all going to be able to reproduce. In contrast, what is the likely fitness of this one female? It's potentially pretty high. There's a high probability she will actually be able to reproduce if she so chooses. In this regard, again, it's better to be the rarer type. So, again, when you're rare, this rare allele has an advantage. As it becomes more and more common, it loses its advantage and eventually can go to a disadvantage. This will maintain genetic variation in a population. And you might guess, if you have two alleles, what would you predict that equilibrium allele frequency to be, if you assume it's symmetric? Well, the answer is 50%. In fact, if it's symmetric, you will tend to see just about always the equilibrium frequency be 1 over the number of alleles. So if you have two alleles, it would be 1/2. What if you were to introduce a third allele into the population? Well, then probably that equilibrium allele frequency would be 1/3. This is assuming a sort of symmetry, which may or may not be accurate, but this gives you some idea of what it could be. Now, frequency-dependent selection is another way of maintaining variation in a population through selection. And we talked about a couple of different modes of single locus selection here. Some of them remove variation, some of them retain variation. Okay, so directional selection favoring one allele, this essentially eliminates variation, right? And the effects of dominance will change the speed with which variation is eliminated, as well as the strength of selection, of course. Heterozygote advantage maintains variation. Heterozygote disadvantage is unstable, so although you can argue that it maintains variation, it only does so under unrealistic circumstances, so it probably will eventually lead to elimination of variation. And negative frequency-dependent selection will maintain variation as well. All of these are affecting genotype and allele frequencies, but they're acting through the phenotype. Now, this discussion, we're looking at everything in the context of the effect of alleles at one gene. Obviously, that's not what Darwin did. And it's obviously not what people do when they're applying artificial selection, right? They're looking at the traits as a whole. They're looking at the phenotype, the variance. So let's tie this back to the principles we talked about earlier in terms of heritability and things like that. Well, we'll start doing that in the next video. Thank you.